The research that we do in my lab is all related to this one area that we're interested in, which is linking up the genome of an organism to either how they behave in nature or what they're doing out there in nature. I've chosen to specifically focus on insects. Insects are fascinating and they're everywhere and they're really easy laboratory animals to work with. If we can understand at a very basic level, the DNA level, what determines how quickly or how slowly an insect grows, we can use that to refine some of these forensic estimates that we make to determine how long somebody's been dead. And that's a little bit dark, but we do that because the insects have evolved to be able to detect the smell of death very, very quickly, so they can act as a clock.
I'm specifically going to be studying how humidity affects their rate of laying eggs. Humidity is used as a factor that influences when a fly arrives at a body. So the presence of fly larvae, maggots, eggs, as well as species is used to calculate a time of death.
If we can understand at the DNA level how an insect is sort of pre-programmed to develop, then we can refine our estimates of these and make it a little bit better. And that's what we want to do, right? We want to get as close to that true time of death as we can, because that helps, of course, with the criminal justice system. We don't want to get it wrong. It turns out the blowfly itself and the way that it acts in nature can be very useful to us humans as well. So it can act as an environmental sensor, or an environmental drone, as we've called it, where it goes out there and it samples the environment. And then we can call it in by using stinky meat, which is our bait, and they all come in, and we can analyze a lot of the information that they're carrying within them to be able to understand things like biodiversity in an environment and how that changes over time.
I'm working with Blowfly Dna, and so blowflies are known to eat feces and decomposing bodies. So our hope is that by analyzing the DNA in their guts, that we can determine what animals they have been feasting on. It will help us determine the biodiversity and the populations that are present in certain environments. We could maybe look at if that was due to human factors or maybe climate change, things that affect the environment like that.
Because the flies are out there in the environment collecting all of this information, they're also picking up a whole bunch of chemical information as well. And one of the projects that we have, that we're working on, is to determine whether or not the blowfly can be used as a chemical weapon sensor. So you don't have to send a human into an environment that has a suspected chemical weapon. The flies are there, the flies are flying through that environment, they're picking up that information, and we can extract it from the flies. Another way that we can apply this that's not forensically relevant is for the growing field of insects as food and feed. A lot of the food sources are unsustainable and are contributing to climate change at alarming levels. A chicken's natural diet is insects. So why aren't we feeding them insects? Instead, we're growing crops to feed chickens, and that's not their natural diet. So it's time that we sort of get back to where we should be. And I will say insects use less land, less water, and produce less emissions per unit of protein produced.
I'm trying to inject a chemical inside a number of beetle larvae, or pupa rather. And our hope is that these chemicals will trigger genome editing events inside the animal. And after the events, the animals would produce offspring that have the genes that we want to change altered, so that their offspring would grow faster or bigger. This project it's essentially wanting to make a feed crop for livestock that are more efficient to produce and more sustainable. This project is utilizing the CRISPR-Cas9 techniques which has become sort of the standard toolkit for projects such as these.
Just being part of a research group is really valuable to be able to learn how other people think, how other people arrive at some of these questions that they get to, and how they design experiments in order to be able to answer those questions. The challenges are really good for you, and it'll set you up for the future and prepare you for what real life looks like, right? Real life is not what's in a lab manual where everything works out perfectly. Real life is where something may have gone wrong and you need to figure out what that was. And you need to be able to apply those critical thinking skills, and you can hone those during research.